This summer, I examined host water stress effects on mealybug vector prevalence and survivorship. I like to start by setting the framework and talking about the big picture or the greater implications of my work. Though my work is focused mainly on grapes and vineyards, there are many important scientific concepts behind the work that can relate to other things. As you can see from the title of my project, water stress is a very important part. So water stress in plants is water deficit or limited water availability. Why is this important? Why should we study water stress other than the more obvious reasons? So interest in plant water stress has been around for quite some time. In 1974, an entomologist named Thomas White used water stress to describe the behavior of looper caterpillars on pine trees. In a paper published in the famous journal Ecologia, he posited, "It is known that water stress produces marked physiological changes. In particular, the total nitrogen and total proportion of soluble nitrogen is increased in leaves. Such increases in the amount and proportions of nitrogenous food might well result in greater chances of survival in insects." So water stress leads to more food, leads to greater survival. Is this true? So Thomas White went on to become a noted professor and the author of several books. But this hypothesis that he posed many years ago remained very influential in water stress research up till today. And my work will deal primarily with talking about this hypothesis. So flash forward a couple decades, and we're facing one of the worst droughts in California history. And we know that climate change has a big role in this, and that it may produce other forms of extreme weather, including droughts, floods, and heat waves. But what we don't know is what kind of effects droughts and other factors caused by climate change may cause on crop system dynamics and other ecosystems. And so, as you can see, water stress is still very relevant today. And so I work with grapes, and some of you may be wondering, why study grapes? So I took this number from the California Department of Agricultural Statistics, listing the top 10 valued commodities in California, and grapes came out to $4 billion. So you can see it was the second on the list. It's a very large, economically beneficial plant, and I hope this is compelling reason enough. The Alameda Lab has been working with water stress in grapes and mealybugs for some time, but primarily in relationship to leaf roll disease, caused by a family of viruses carried by mealybugs through plant to plant. And we did find that there was a correlation and there was a trend caused by water stress between both mealybug acquisition and inoculation, which is picking up the virus and transmitting the virus to other plants in relationship to water stress. And this is what inspired me to do my work today. Though I was moving more away from the pathogen itself and more towards the driving factors of the spread of pathogen, which is the mealybug itself. So I aim to focus primarily on how does plant water stress affect the movement and survivorship of mealybugs on grapes, which are both important factors in pathogen spread. And so I'll talk a little more about the host vector system that I'm working with. And by host, we mean the plant, and vector is the insect that transmits viruses. So our host, I use grapes, a variety called Pinot Noir. And Pinot Noir is a commonly used red variety in winemaking. And we typically use Pinot Noir in our lab because it is a great indicator of leaf roll disease symptoms. And so for my work today, I continue to use this variety to remain consistent with our previous work. And so mealybugs are a very common vineyard pest. And though they are quite small, they can do serious economic damage in clusters. And if you can imagine, one adult female can produce an ovisac of hundreds of future mealybugs. And so that can be quite devastating. And so the particular species I used was the vine mealybug, or Planococcus ficus, because it is a very invasive pest in California. And it is also very noted for its high levels of movement, which is important for my work. So now I'm going to talk a little bit more about my experience. So in order to answer the question behind my entire project, I divided it into two different kinds of experiments. The first, which I'll talk about first, is the mealybug preference, which aims to describe the movement aspect. And it was short-term pair choice experiments that looked a bit like this. And so the main premise behind this was having pairs and giving the mealybugs a choice between the two pairs. And the two pairs would have different levels of stress. And so to get started, we had to water stress the plant. This involved 4 different treatments designed to give different levels of stress. And the first treatment, which we called high water treatment, was aimed to give low stress. And it involved 200 milliliters of water into the plant daily. And it included this dish tray on the bottom of the plant to collect water so that there was a continuous source of water in the plant. The second treatment, which was aimed to get medium levels of stress, did not involve the dish tray, but we took the plants off water for 2 days, and then we top watered for 4 days with 20 mils. And then below, we took the plants off 3 days, and then top watered them with 10 mils. And so the final treatment, which goes back to our plant water stress hypothesis, this hypothesis actually snowballed a second hypothesis later on called the pulse stress hypothesis, proposed by Huberty and Deno in 2004. And Huberty and Deno refuted the plant water stress hypothesis using meta-analysis and claimed that it wasn't water stress that provided nitrogenous food and greater survival to insects, but it was intermittent water stress. And so we strove to replicate this by having a set of plants that we treated like low up until the experiment, but right before we started, we added 200 mils in a flush of high. So after we water stressed the plants, we had to verify the stress levels. And the measurements we used were midday leaf water potential. And this is a very common technique used in vineyards today. And what it involves is a pressure chamber attached to a gas tank with usually nitrogen, since it doesn't react with the plant. And what we do is we place the leaf of the particular plant that we're measuring, and we measure the amount of gas it takes to push water from the leaf to the petiole. And there's a correlation between the amount of pressure it takes and the water stress of the plant. So after we've collected that, we would pair the plants, and it would always be one well-watered plant, one high water treatment plant, with a variably stressed plant. And that could be low, medium, high, or flush, which is not pictured here. And after that, we would place one bug on the variably stressed plant and then observe its movement to determine which choice it would take between the 2 plants. And so our initial experiments determined that it took about 3 days for these bugs to settle, and that informed our subsequent experiments, and we would only observe after 3 days. And so that was my mealybug preference experiment. The second part of my experiment is mealybug survivorship, which were long-term observational experiments, which looked a bit like this. They were kept in growth chambers and observed every 2 days and watered every day, with a similar method of starting with water stressing the plants. And then after that, we would place 20 bugs on each of the plants and observe from the first stages of their life cycle all the way until the next emerging generation, which took about 55 days for our first replicate. And so after we collected all this data, we did some analysis, and we determined that indeed the water treatments that we provided the plants did have an effect on their water stress, and it was statistically relevant, and the treatments were all different. We also found that water stress had a significant effect on insect movement, which is what we believe. And so this graph shows dispersal from the initial plant. On the y-axis, a 1 signifies a movement away from the initial plant, and on the x-axis is a measurement of water potential, where on the leftmost side, a lower water potential indicates less stress, and as you move towards the right, it's more stress. So as you can see, in the higher regions of stress, there's more dispersal. We can also break this down into the different levels of treatments and include some data points that we didn't have water potential measurements for. And though we're not entirely sure the difference between the treatments, we can see here the cluster of the high water treatment where there was very little movement. And so we can say that lower water treatment led to more dispersal. And so our survivorship experiment is still in progress, so all the data here is preliminary. We did find that water treatments had a significant effect on plant growth, but we're not entirely sure about the nitrogen or other nutrient content of the plants, and we hope to do more analysis on that later. We believe that more instars transition to adult stage in higher water treatments. And so this is proportion of instars moving to the next stage, and you can see here, the higher primary water treatments had more. We're not entirely sure how this plays into things, but there are possible variations in both width and length of mealybugs among treatments, as well as ovisac size. But we still need to do more statistical analysis to determine if that's true. So going back to the plant water stress hypothesis, is it valid? Our experiment does not support it, and is consistent with Huberty and Demno and other current literature. In terms of pulse stress hypothesis, we may neither confirm nor deny, but we will do more work and hopefully be able to give a better response. So future implications of our work: high host water stress may cause increased vector dispersal and concomitantly increased virus spread. So this is a very, very awful conclusion, because with the droughts, that means there's more mealybug movement, more virus spread, less quality wine. The world is ending, right? But there's a silver lining to this, and this is what we think we know from our current work. So our past work gave us an idea that it was supported that it's possible that on stressed plants, mealybugs were more likely to pick up viruses, but on stressed plants as well, they were less likely to infect uninfected. So our work would be that they're also more likely to move away from stressed plants. So this means that it's possible that management techniques in already irrigated vineyards can focus on reducing water treatments to uninfected crops, so that the mealybugs will not be able to infect it as easily or prefer to stay on them. So that's very economical, especially with our limited water availability and continuing droughts. So hopefully that is supported. So science is a collaborative project, and of course I couldn't have done this project by myself. So I'd like to thank a couple of people who've helped make this happen. Rodrigo Almeida, my faculty PI, who's the main reason for me being in the lab and doing this work today. Kai Blaisdell, who has been giving great insight and feedback throughout the project and has supported me from the conception all the way to moderating my talk today, and of course much more. And Tino Istro, my greenhouse manager, who supplied the majority of my project. Mathilde Kabi, who is sitting in the audience over there, and who has done much of the data collection for the survivorship experiment. And of course, the numerous advisors that I contacted for my methods, as well as the SERP and Rose Hills Foundation for giving me this opportunity. Thank you.
Excellent talk, great project. So we have a couple minutes. If you have a question for Megan about this project, do we have any questions at all from the group? Yeah.
So I think it's really cool that the mealybugs will prefer the plants less, you give them less water, but to what extent can you reduce the water treatment and not kill, well, because it's the $4 billion deficit due to the mealybugs, can that be improved by reducing the amount of water? Because wouldn't the grapes be less big, and wouldn't that still hurt the plants?
That's true. That's a good question, actually. I'm not entirely sure to what extent would keep the quality of the grapes as well, but that future work can definitely focus on working on that and finding a good midpoint of reducing water treatments while keeping quality high.
Yeah.
To sort of follow on that, I know that wine people can be really picky about the amount that the grapes were watered. So do you think that that could pose an obstacle to implementation of like some of the advances that your findings suggest?
This is definitely true, but I do know that currently in wineries today that there are differences between the amount of water that they've been getting already. So the wines that you're drinking, they're not necessarily like, oh, I can tell that the grapes haven't been watered.
They say they can.
They say they can. I'd like to talk to them. I don't really know if that's true, but yeah, definitely there can be more work done in that area too. But yeah.
Any final questions at all? Hey, well, great talk. Thank you.